harnessing the energy from the sea areas. It's a very complicated economic sector, but that is worldwide in a growth.
Good morning, ladies and gentlemen. Welcome to the Vliz Marine Science Day 2022. I'm Hetty Helsmortel and it's my pleasure to be your host today. Due to the pandemic, this is once again an online event like it was last year, but no worries, we'll make it an exciting and fascinating morning with a mix of live interviews and pre-recorded talks, so that will be very, very nice to follow. Luckily, completely live here with me today to officially open the Marine Science Day is Jan Mees. Jan is the director of the Flanders Marine Institute. Jan, welcome.
Thank you.
2022, I think quite an important year for Vliz. I heard there is a new covenant approved for a period of 4 years, I think.
5 years.
5 years.
Yes.
For people who are not familiar with the concept of covenants, what are these things? And maybe more important, what impact will it have on marine research in the years to come?
Yeah, Vliz is evaluated every 5 years.
Okay.
And basically this results in an advice to the government, the Flemish government, but also the province of West Flanders, on whether to continue to invest in Vliz, in marine science in Flanders, and what direction we should take in the future. Our evaluation that was carried out last year was very positive. So that means that in general we will continue as we were. We will continue to provide science supporting services to the wider Flemish marine research community. We will perform our own research, and we will make sure that we invest in the valorization of our research towards different target groups.
Okay.
New is that there will be, amongst others, a focus next to research that valorizes towards the blue economy.
What's the blue economy?
Blue economy is the wider domain, different subdomains actually, of economic activities that have the sea as a common playground.
Okay.
So you have to think about coastal defense, but also blue biotechnology, pharmaceuticals from the sea, sustainable energy, harnessing the energy from the sea areas. It's a very complicated economic sector, but that is worldwide in a growth mode. We expect growth figures of more than 10% for the blue economy worldwide, and also Flanders has decided a couple of years ago to really invest in this. We have a blue cluster that is well established, a cluster of blue companies, but Vliz is mandated to make sure that there is an input of research through innovation pipelines into the blue economy.
Oh, that's really interesting. And that was not the case before?
That was already the case, but this mandate is reconfirmed and we will have to invest more in this. And next to that, there is a shift of focus to large research areas that the Flemish government thinks will be very important globally, but also for Flemish society in the next years. I want to mention 3 of them. First of all, there's the relationship between the ocean and human health. It will be a very important point of focus for Vliz, but actually it's very broad. It goes from effects of pollution to mental blue gym aspects of the seaside presence, also the physiological effects of sea air, for example, on human beings. The health benefits is something we will study. Next to that, another large area will be everything about climate, the ocean climate nexus. And more specifically, the ocean climate biodiversity nexus is something that we will heavily invest in in the next years. We will refocus part of our research towards climate research.
Okay.
And that's something that's very important for a society like ours, of course. Not only the adaptation that we have to organize, we know that the sea level rise is coming, it's unstoppable, but also mitigating research, looking into ocean-related solutions towards climate change. Also, we are certain of that there is an important spillover towards the blue economy. There are opportunities there. If we smartly invest in innovative, rich areas of the blue economy, I'm sure that Flanders can make a difference worldwide.
And the third one you wanted to mention?
The third one I wanted to mention is the valorization of our infrastructure and our databases. So infrastructure and data-driven research will be very important for Vliz. Over the past 10 years, basically, we have invested a lot in high-end, cutting-edge infrastructure to do oceanographic research. We have invested robotics.
Robotics, yeah.
Robotics center was established in the last covenant period. Basically, it was a one-off investment. We now have investment budgets to continuously reinvest in robotics, have additional robots in our collection. But these have to be valorized, of course. We have to use them, and we will make sure that we make them operate in the world's seas to the benefit of climate research again.
Wow, a lot of work to do.
A lot of work to do, and also we have, from the beginning of Vliz, we had a mandate to collect, integrate and redistribute scientific data, oceanographic data. These collections are huge now. We have a very important data center.
20 years of research.
20 years of research, but also data archaeology. We've been translating old data to modern carriers. Not only Belgian data, but European data, global data. And now is the time to move on from this phase of analysis and collection towards a phase of redistribution, valorization, integration, synthesis, basically, to make sure that we valorize the data capital towards new and exciting science.
Yeah, sure. Jan today has a focus on young researchers. Many young researchers will present their work here. Why is today's event so important in the grand scheme of things for marine research in Belgium, but also worldwide, maybe?
Yeah, we need a new talent. We need it. We need a lot of people. You know that last year the UN Decade of Ocean Science for Sustainable Development has started.
True.
The plans are now in place, slowed down by COVID, of course, but the framework is there. And we expect from next year onwards that this will really take off with a lot of international programs, actions, projects that will start, that will trickle down to Flanders. And we will do our bit. We want to contribute to the UN Decade. And we need a lot of people to do that.
Wow.
So it are very exciting times to be involved in marine science. And we need young scientists of all disciplines to get involved. Even people that can contribute to marine but don't know it yet, with their specific skills, tools and...
like biologists, chemists?
That are the trivial ones. But you also have the social scientists, the historians, the not so trivial ocean scientists that we really want to get involved. We need these transformative aspects of interdisciplinarity that we want to have, and that's why a day like this is so important, that it's without too many barriers, people can get informed about how exciting ocean science is and what the opportunities are in the next few years to contribute.
Wow. Jan, thank you very much for officially opening the Vliz Marine Science Day. We will see you back around noon to close the day. Yes. So I would say, in the meantime, sit back, relax, and enjoy all the stuff the young researchers want to tell us. All right. Before we move on to our first keynote speaker, I have two practicalities I want to share with you. First of all, if you have any questions or remarks or ideas, we have a chat box. So please use it. People of the organizing committee are available to answer. That's a really nice tool in these online event areas and days to have someone in person chatting with you. So please use it if you want to use it. Second of all, also this year you can vote for the best poster. So we again have a poster session. It's again a digital poster session, but you can vote for the best poster. You can, by the way, do that until the end of today. And I heard that some lucky voters will also get a special gift, so to stimulate everyone to vote. Yes, and then it's time for our keynote speaker. And wow, this year it's really someone with name and fame. It is Patrick Sorgeloos. Patrick is a retired professor at Ghent University. But he's one of the most important experts worldwide in the research field of aquaculture. And today Patrick will talk about his life, but also his work as a renowned authority, and also about one of his favorite aquatic creatures, and that's the brine shrimp. So I would say please be all ears for the very interesting story Patrick wants to share with us.
Thank you, Hetty. Thank you, Jan and Vliz, for this kind invitation. Dear participants in this Vliz Marine Science Day, in the following presentation, I like to take you on a scientific journey of just over 50 years with one fil rouge, that remarkable crustacean, the brine shrimp Artemia. A story of coincidences, luck, challenges, opportunities, but especially a most rewarding experience of so much positive interaction with hundreds of good colleagues and friends all over the world. It all started in the late 1960s when I was looking for a thesis subject. And it was in the lab of Professor Foutré, embryology, that I met with Lieve Kriel. And they were using the brine shrimp Artemia in embryology research. And they had one problem. It took them 2 to 3 weeks to grow the Artemia up to the adult stage. And only then they could start their experiments. And often they told me, often 2-3 days before they have to start the experiments, the animals die. A perfect subject for a licentiate thesis study would be a standard method to culture Artemia. Under a very good leadership of Professor Guido Persone, I started in the lab of Professor Vliz, lab of ecology, to develop a technique following what you see here on the screen, the life cycle of the brine shrimp Artemia. I graduated in '72, got a scholarship to go for a postgraduate, part of the postgraduate at Duke University Marine Lab in the US. And interesting surprise there. The two scientists, Buchhout and Kostlow, very famous for their work on the embryological development of crabs, crustaceans in general, but particularly crabs. And I heard there for the first time that they were using Artemia to feed these plankton samples so that the crab stages, Zoea, Megalopa, those among you familiar with crab morphology will know that these are very, very different stages going through the metamorphosis. And both scientists told me in Japan, they are starting aquaculture and Artemia is used as a food source. So first coincidence, first change in my career, reorientation of my PhD study, Artemia as food in aquaculture. So in the catacombs, some of you might recall, in the catacombs of Ghent University in the Rosier, we started working on the different aspects of the biology of Artemia. Rather complicated problem with the cysts, these embryos that can be stored for a long time, and that in fact you can use, I like to call it magic powder, that you can convert within 24 hours to a substitute for plankton. And then, as we see here, use it as a kind of artificial plankton, as food for baby fish, baby crab, baby shrimp, you name it. That was at the time that brine shrimp Artemia, under the form of cysts, was available from only 2 sources in the United States, the Great Salt Lake and the salt works in the San Francisco Bay. And as you can see, less than 100 kilograms were used on an annual basis. Why? Because it was only used for aquarium hobbyists to feed their guppies, maybe to feed their seahorses. 1975, I got my PhD. the opportunity of my life, 1976. I'm invited as the Belgian representative, can you imagine, as a 20-year-old scientist, invited to represent Belgium at the first UN FAO technical conference on aquaculture. And big surprise, in the opening address, the chairperson is saying, there's no future for Artemia, we cannot develop aquaculture, say in the Philippines, say in any country in the south, and we are dependent on hard currency, on US dollars, to buy the food for the food to grow then the fish that we want to culture for the poor people. I raised my hand. I dare to do things that today you don't do at the UN conference. And I said, the situation is artificial. And look, there are many other places where you can harvest Artemia and we can improve. So I was turning a challenge, in fact, into an opportunity. And apparently I was able to convince FAO because I got the opportunity to spend several study visits at a very interesting center in the Philippines. And no surprise, where you think, oh, I have a PhD, I have developed a number of new techniques, and the first thing that I had to admit and realize there, my goodness, what I developed in Ghent is not necessarily applicable in a context of a developing country. Just one example, we were growing Artemia on spirulina that you had to import. And people in the Philippines at that institute, they told me, very sorry, but we will not use again US dollars to buy the food, as I was just explaining. So either back to Belgium, or how can I find a solution? And I was asking the people there, please give me whatever agricultural waste you can think of. Let me try and see. And that's where, surprise, surprise, we found out that rice bran, very, very limited food value, but a very good source of food for Artemia. We learned more about the biology, see the reproductive cycle, how to produce first a big population of Artemia, what we call the Artemia biomass, and then how to switch these animals to let the mother Artemia switch to these dried cysts production. We had done, let's say, the pilot testing in the Philippines, got the chance to verify our hypothesis, again, with great luck, in one of the biggest regions of salt production in the Nordeste in Brazil. Can you imagine? 250 grams of Artemia were introduced in April, and I think it was in August or in September that the first metric ton of cyst was harvested. Same, another FAO project at that time in Thailand. You see the cover here of the Fisheries Gazette in 1977, where we were able to demonstrate that in local salt farms during the dry season, because in the rainy season Artemia cannot survive, and you can produce Artemia. So within a couple of years, we were really fortunate that we could convince FAO that there was a future for Artemia, provided, of course, that the things would be further followed up. And FAO was recommending us to set up the so-called Artemia Reference Center at Ghent University. I quickly realized that the challenges were enormous, because what we had promised in Kyoto, we had now to put this into practice. And I quickly realized this is not what you can do in a small lab with just a few people. So we set up the International Study on Artemia with colleagues working on Artemia in Greece, in the UK, in Spain, in Italy, and the United States, 2 labs in the United States. Organized a number of training courses. This was the first one here in Ghent, but training courses in a couple of Asian countries, also in Mexico and in Brazil. And that's how you see just a flavor of some of the papers and books that have been published. But under the common heading, as you can see here in the slide, under the common heading of International Study on Artemia, we published over 60 papers in peer-reviewed journals. Conferences were organized, Corpus Christi, Texas, Antwerp, an FAO manual, a number of other books were published, and okay, we were able to see the ball rolling, Artemia being used as a successful food source in the hatcheries, be it for marine fish, even some freshwater fish, crab, prawn, shrimp, you name it. And then not surprising if that hatchery aquaculture can develop, that the demand, and lucky enough also the provision of Artemia cysts could follow. And you see there the figures today. The world market, the world consumption of Artemia is in the order of 3,500 to 4,000 metric tons on an annual basis. And in the meantime, that hatchery sector valued at a couple of billion US dollars. What is more interesting to see here is that it is responsible for a final production of over 10 million tons of high-valued aquatic products. 1983 was another particular year. It was at that time under the impulse of the Flemish Investment Society, the HIMF, that some of you might remember Plant Genetic Systems. In the same year we were setting up a smaller company, of course, Artemia Systems, which eventually, you see, 1991 was taken over by Invi Aquaculture and is still one of the leading companies in the baby food, I could call it, in aquaculture. Going a bit back now on what are these different interdisciplinary, multidisciplinary, multi-stakeholder aspects in such a career. And I think the responsibility of a university researcher or professor is to focus on the three pillars: education, research, and service to society, or as it is called here, outreach. 1991, very crucial year again in our work, was in fact under the suggestion, upon suggestion of the late rector De Meyer, who said, yeah, but you are doing good work in research and with developing countries, get more involved in education, set up a master program. We didn't feel that we had the competence, honestly speaking, to cover all the aspects of aquaculture. And thanks to good friendship with friends in Wageningen University in the Netherlands, we set up a master program in Ghent that was, let's say, mirrored to some extent with the master program in Wageningen. So our students went for a full semester to Wageningen, and the Wageningen students came for a full semester to Ghent. This has been a very rewarding experience, and also one where we quickly involved the contacts we had in the south, and you see the arrows go to Africa, Asia, Latin America, and where we did our best to send most of the MSc students for an internship, for thesis work, to the places where they had a finger on the pulse in aquaculture. Maybe they could get good training in Belgium, but this is not where you see the practice. So that's where we tried to combine the two. Of course, research-based education, that implies that you also have to see that you focus on the research. And maybe you heard our specialty is Artemia and larviculture. This is not the basis for a full master program. So that's where we were very fortunate that we could convince, not always easy, convince colleagues, specialists in virology and bacteriology and in different other faculties. And I'm very proud that we have that UGent R&D consortium where you see the different specializations, the different disciplines, and really a good example of interdisciplinary cooperation. And of course that means that you do the research with the different partners in Ghent and colleagues abroad, but that you also look into proper technology transfer, knowledge transfer. And that eventually helps us, and is in fact a reality today, that we can do a lot of joint research, even at the same level in the north and in the south. And in the south, where you have much more activities with Artemia, with aquaculture. So bilateral agreements, working together with the farmers, working also here with the sector in Belgium. And a number of companies, you see the names there, could benefit from either the students that we were training or the contacts that we had, and help, for example, a company like Colruyt to do quality control through some of the Vietnamese students that had been trained here in Belgium and went back to their country. I think a number of you know that Vietnam has been a very peculiar case in our career, 1982. That was, I can tell you, under very difficult conditions just a few years after the war. But this has been a fantastic experience where you see the comparison here 40 years ago and today, the poor life of the salt farmers that today have been able to integrate salt production with an aquaculture production with very significant socioeconomic benefits. I mentioned already research is interdisciplinary. Larviculture is a particular specialty in the field of aquaculture. So it was also in that famous year 1991, a lot of things happened in that year, that we started, and maybe in the front row you recognize some people from, at that time, ISWO, later Vliz, where we were organizing together these larvae conferences, which, as you see now, is really a very popular event in Ghent every 4 to 5 years. It also helped us along the way to go more into fundamental work. And no time to go into the details, but the gnotobiotic Artemia system has been a unique new tool in Artemia research, where in axenic conditions, bacteria-free conditions, we can culture Artemia, and that gives us a lot of opportunities to do different research aspects summarized here in this scheme. We started developing that, I think, at the turn of the century, and then with the coming of Peter Bossier, it could be further developed into a system that I cannot remember if it is 15 or 20 PhDs that could be accomplished using that gnotobiotic system. Again, no time to go into the details, but I'm proud to say that today Artemia is a real model organism in quite a number of disciplines. I will not go in the details of the ones that you see there, except the last one, the cell cycle studies. Remember that my work started with the work of Lieve Kriel and Professor Foutré on embryology and what might be the mechanism to start and stop the metabolism. A colleague in China has now identified the gene in Artemia that is responsible for the switching on and off of the embryonic development. He has transferred that gene in cancer cell lines, and he can switch on and off cancer cell lines with that gene of Artemia. And okay, the good news is that since last year we completed the annotation of the Artemia genome, so an extra tool becomes available to continue that basic work. I take the opportunity to show this slide here and say this is all the result of good teamwork. And teamwork where you motivate the people. If you have highly motivated staff, you accomplish so much more than a couple of individuals who might do their best to do good research. Okay, I think you start to realize that we have been heavily involved in networking, so not only what you see to the left in a number of European organizations, but with the contacts that we had and that some of our European colleagues didn't have in Vietnam, in Thailand, in a number of countries. So we have been involved in a lot of such extensions, and the one I'm most proud of is the one that was just completed under Horizon 2020 a couple of years ago, where I will not cite the names, but Vietnam, Thailand, Bangladesh, Malaysia, very successful multi-stakeholder platforms have been set up in these countries. China, another country where first visit in 1985, we have been able really to develop good relationship with institutes and famous research organizations, and you see with good project funding from quite a number of organizations. And we brought, I must say, quite a number of our European colleagues, see at the bottom of the slide, and some of you might recognize Jan Mees, the director of Vliz, in the picture when we had one of these exchange visits to Qingdao in, if I remember well, 2011. I want to spend 2 more slides on China, because what has impressed me so much from these early visits in the 80s is that holistic approach, that polyculture approach in aquaculture. And it's so good to see that what is a common technique for already a number of decades, where you see, in fact, these are the old slides that I'm showing you with the wooden cages for fish, and in the back you see the different long lines for the mollusks, different species, for the seaweeds, different species, a fully integrated approach that, in my opinion, is something that we in the West, where we have focused, and I will not criticize, we have focused on monoculture. This is more the Western approach. Zoom in on one species, try to get the best out of it, make it the most successful. But I'm very much convinced that now the time has come, and when we hear about the EU and the circular economy, this is what we need to put in practice. We have to convert or adjust that business aquaculture towards a food aquaculture. That means integrate with other activities, not only in aquaculture, but even in other industries. So maybe an old slide that I was using 15 and more years ago to convince, I remember the meeting here in Ostend, to convince some of the the companies that were early in the windmill parks, and that they were very hesitating to see a possible integration with aquaculture. Very good to see that there is now such good activity in the North Sea, as an example, in fact, for a number of other countries in the world. So, towards the end of the presentation, I like to give some hints for the future. Traditional capacity building in the North, very good. I think we have accomplished a lot in the Western world with EU funding, with FAO funding, to help a number of partners in the South. I think we should more focus now on joint capacity building. Joint capacity building where we have partners in the South. Remember some of our graduates of 20 and 25 years ago who were all in leading positions, not only in education, but also at the government, Vietnam, Indonesia, to mention these two, but also in industry. And that proud to show here the first PhD double degree between Chulalongkorn University in Thailand and Ghent University. And in order to stimulate such a program, Ghent University, I'm very pleased to mention here that in the name of my wife, the Ghent University set up the Mama Magda Fund in order to facilitate, to provide that seed money to complete the 6-month research, which is a minimum requirement to go for the PhD double degree. Let's go back to the fil rouge and finish here with Artemia. An Artemia reference center at Ghent University, yes, but see, in the meantime, FAO has recognized one in China, the Asian Regional Artemia Reference Center. And 2 years ago, and you see that quite a lot of partners from the public and the private sector are involved in a new initiative, what we call the International Artemia Aquaculture Consortium, and where it is the intention, and very pleased that FAO was organizing such an SDG-aligned Artemia aquaculture workshop at the Global Conference on Aquaculture last year. No time to go into the details, but where FAO wants to have a review and see for the decades to come what is needed in order to protect Artemia, assure that there is good sustainable harvesting, and look here on the slide, the opportunities in maybe a number of other continents for such socioeconomic activities. I want to finish just with some brief topics. Look and take up opportunities. Don't hesitate to take risks. Fall and rise. Maybe you are surprised to see the sour and the sweet apple. If an apple is always sweet, you do not appreciate. Time by time you need to have a sour apple. You understand what I mean. not every day, not every time it will be as lucky and as good as you are expecting. Passion for your work. Don't give up. Persistence. Motivate and encourage the people around you. Very important. Just make sure that it's the team that is committed to work. And give and take. So exchange information, maybe even before it is published. That's the way you can motivate people. Don't hesitate to take risks. Ask advice. I'm still thinking about the word wisdom. I'm still asking advice to people that are older than me. You can always learn from them. Enjoy your work. And last but not least, find a good balance with your family life. And in my case, this is with the 11 grandchildren that you see there in the picture. Thank you very much.
Thank you, Patrick. What a life story. Wow. I would like to listen a whole day to you. It's really impressive. It's really impressive. Maybe if we go back one slide, I think now that a lot of young researchers are listening to us and watching us, can I take a few of them and ask you, let me start, making mistakes. I think that's a really interesting one. What are mistakes that you learned from?
Well, and this is an interesting question because just 2 weeks ago I was giving a presentation in Ecuador and I was the scientist who stand up to say I have made a mistake. Many people were looking out, what is he saying now? Well, I cannot go into a lot of details, but in 1985 we thought that this was a great discovery, a method, the decapsulation method it's called, with hypochlorite and sodium hydroxide to remove the shell of Artemia. Okay. This technique has been widely applied, successfully applied, until we started to realize hypochlorite, sodium hydroxide, this is not environmental friendly, and we should have considered that at that time. Lucky enough, we are now working on alternatives, but this is an example where you have, also as a scientist, you have to be honest with yourself and at a certain moment even admit, sorry, I made a mistake.
And what were the reactions?
Well, a surprise, because they were still applying in Ecuador that technique. And they are one of the few countries in many, many other parts of the world, decapsulation is no longer applied.
Thanks to you saying I made a mistake.
Yeah, well, and it had such an impression that they said, why didn't we know earlier? Yeah.
Communication is really important.
Yeah.
I was very happy to see your slide with education, research and outreach. Do you think these three things, that has to be something one single academic has to do, or do you think it's more on a group level? Some of them focus on research, some of them focus more on communication, valorization, others do more education, or would you really stimulate everyone to take the three things together?
Yes, but I can imagine that, okay, then one might specialize a bit more on outreach, but I think it's crucial for a good teacher, for a good scientist, to cover the three.
Okay.
First of all, today education at the higher level is research-based education. Sure. Second, and I gave the example for my experience in the Philippines, I can give you many other examples where the link with the industry, the link with the farmers, we don't have the time, but I could tell you so many stories about mistakes that I made in convincing farmers in Thailand, you don't have to apply that green technique. The arguments that they were giving were not correct, but I couldn't blame the farmers that they were giving the wrong argument. And I said, you can achieve exactly the same in clear water. Today we know that green water is microbial stability.
So they were right.
They were right. And so it is that interaction, and I always tell the scientists, don't sit in your ivory tower. And also don't wait, like I did with my PhD, to develop a technology, be very proud and publish the paper, and then when you go to the farmers that they say, hey, very sorry, but this or that does not work.
Yeah, so go out there and talk to them. Yeah, it's really nice. I don't know if it's here, but I think the Asian-China influence is also really interesting. Do they have a different approach towards life? And you mentioned the holistic thing.
I like that question.
Okay.
Well, you say the word. For me, Asians, they think more and they act more holistic than we do. Ooh, that's maybe a strong statement. I will focus first now on Europe, I will say the Western world. In the Western world, and that's also why we do monoculture in the Western world, we zoom in on a problem. And we really pay all the attention. If there is something that, let's say, is a sideline, okay, this is maybe to a wastewater treatment plant. No, let's see this as a sideline. Asians, somewhere, and many times we as Europeans, we consider this as a criticism, that we say, oh, this is intuition. I think there could be a good synergy if we could understand and see how in science and in education that Asian mentality, perspective, and the Western Descartes approach, I think that could be a win-win.
Did intuition play a crucial role in your research career? Would you advise young researchers to follow their gut?
Yes. When you get an idea, definitely yes, but use the good scientific principles to verify your hypothesis.
Yes, that's in these times of anti-vaccine, that is really important. Ask advice. Even you still ask advice?
Yes.
To people older than you are?
For me, there is such an important word, wisdom. And wisdom comes with the years. And that's where I see also a huge difference between that Asian approach and our Western approach. Our Western approach, and I fully appreciate, you turn 65, don't take responsibility. I explain it a slightly different way. Don't take responsibility anymore. It's the next generation. Fully agree. If some of these people are still motivated and interested to assist in research, attracting funding, even giving some classes, why not?
Well, it was a pleasure and an honor to meet such a wise man. Thank you very much, Patrick, for being here today.
With big pleasure, and wishing all the young scientists not only a good day, but a very good career. Thank you.
Thank you. Live here with us today is Martien van der Moortelen. She is a live scriber. Martien, live scribing, it means that you harvest auditive information, so you listen to talks to professors and you make it visual. And really, I saw you doing it live, and I think it's impressive. A professor was already 4 or 5 sentences away, and and still it's all there. I think, well done.
Thank you.
Why and when did you start doing this?
Well, I started to do this in 2009.
Okay.
I found a book, and I found it very interesting. I found an American book. I found it very interesting. I said, I want to do that. And it was about visualizing vision, mission, strategy meetings from companies.
Okay.
So you call it live scribe. Some people call it also visual facilitation or graphic recording. There are a lot of names. And I called my company Visual Harvesting. You named it already, harvesting. And it's like listening and bring it to the paper in a visual way.
Yeah.
So it could be a visual summary on one page.
Do you think it helps you keep focus? Because sometimes talks 20 minutes, half an hour, for normal people, it's quite normal to lose focus sometimes, but you were really, you got everything. Is this a way to keep focus?
Absolutely.
Okay.
And I would encourage people to do it also,
but
to keep focus.
I can't paint like this.
Well, you know, I started in 2009 and I do it a lot of times, so I'm very quick, but you can start it. And it's about capturing the ideas and if needed, a drawing. A lot of people come to me and they say, oh, the drawing. But the drawing is not the most important part of this work, as I see it. It's about capturing the essence.
So the keywords that are there.
Essence, and maybe the flow in the story. So these things, and the drawings can help. And you can learn it very fast when you do
really?
it. Absolutely, yes.
Okay. And is it more a tool for people who already attended the meeting or the talk to recap what they heard? Or is it also, if you didn't follow the presentation, to see or to grasp? Is it
In my opinion, when you do really the live work,
Yes.
it's better for the people that were in the room or attended the talk. You could do something similar, but then maybe the drawing needs to be a little bit more elaborated. Then you explain things to people that weren't there. For instance, when I say here, Mama Magda Fund, people will remember. And they will remember also why, but other people, they really didn't know. So, but you can explain that. But that's also the reference, what I call between process and product. For me, this kind of life work is process-oriented. And when you go to product-oriented, you're more in the field of illustration.
Well, thank you, Martin. You will stay here all morning. You will also live scribe other talks.
Absolutely,
so, well, have fun and good luck with that.
Thank you.
Next on are 5 presentations by pre-doctoral researchers. Jolien Buijsen, Jadon Beerland, Astrid Rombaus, Steven Pint and Nanou Goedevrouw, they all recorded a talk of 5 minutes for us. And I mentioned earlier that you can vote for the best poster, but you can also vote for the best presentation. So please sit back, relax and enjoy the ride.
Offshore wind farms are built at enormous rates in European waters to meet with the renewable energy targets set by the European Union. They do not only provide green energy, but research has also revealed some positive influences on fish in their surrounding area. For example, fisheries activities are not allowed in most wind farms, and so they can act as a kind of refuge areas for fish. Moreover, species that are typically associated with hard substrate, such as cod and pouting, are found in much higher densities around the steel turbines and the scow protection layer. Such scow protection is normally used to prevent the sand from eroding, but it also offers increased food and shelter opportunities, which is why these species are attracted in the first place. What about species that normally prefer a sandy habitat, such as plaice? Well, that is exactly the question that I wanted to answer within my PhD, because we simply had no idea. So, how did we start? Well, first we asked ourselves if the spatial distribution of plaice is influenced by the presence of a wind farm. And we wanted to study this on a wind farm scale, which comes down to, do we find an equal density of fish within and outside the wind farm, and on a turbine scale by studying if plaice is attracted or rather avoided the hard substrate around the wind turbines. Now, how did we do this? For the wind farm scale, we followed the Bucky approach, taking beam trawl samples within and outside 2 Belgian wind farms, Seapower and Belwind, and this before and after their construction. Using a statistical model, we could show a positive wind farm effect and a higher abundance of plaice within the wind farm for Seapower, but not for Belwind. It is as possible that a wind farm does have a refuge effect on plaice, but that environmental conditions also can play a huge role. For studying plaice distribution on the turbine scale, we performed several diving transects in the Belwind wind farm, and this over the scour protection layer and the surrounding bare sand, by counting all of the plaice individuals that were observed along the transect. Interestingly, up to 4 times higher abundances of plaice were found on the sandy patches in between the rocks compared to the surrounding sand. It appears that plaice is thus attracted towards the hard substrate, but they were only found on the sand in between the rocks. Their presence could therefore be explained by a higher availability of food and their natural burrowing behavior, which is only possible if the rocks of the scour protection are not stacked on top of each other but leave open patches of sand in between them. Now that we knew that plaice distribution was indeed influenced by the presence of a wind farm, our next question was, what are they doing there? And so we studied their diet, and specifically if it was different for fish found near the hard substrate and in the wind farm compared to outside. And so we performed stomach and gut content analysis together with muscle samples for fatty acid analysis, and this of 4 different groups in and around the Belmont wind farm. The first group was composed of animals that were caught on the scalp protection layer near the turbines. A second group was caught in between the turbines within the wind farm on the sand. A third group was caught just outside the wind farm, and a fourth group was caught further away from the wind farm. A multivariate analysis showed a significantly different diet for the group that was caught on the scalp protection layer, and when you look at the species that make up the majority of their diet, you see that they are typically hard substrate associated organisms, usually found on the turbine and the rocks of the scalp protection. A multivariate analysis of the fatty acid profiles, which can tell us something about diets on a longer time scale, showed that this turbine group was different from the groups caught outside the wind farm, but not from the wind farm group itself. So apparently the diet of place is affected by the presence of hard substrate, and they even feed there for long periods of time, of even weeks up to months. Finally, we wanted to know whether this difference in diet also led to a difference in condition. And although the Fulton index, which is based on weight and length of the fish, was higher for the turbine group, this was mainly due to a higher weight of the digestive tract, suggesting that they did have a higher food intake rather than a higher weight of the muscle of the fish. In conclusion, offshore wind farms can affect soft sediment fish species such as plaice in various ways, such as their distribution, their diet, and their food availability. Thank you.
Climate change is affecting countries in the Two Seas region faster and more extreme than previously expected. The countries in this region face a major threat from increasing sea levels, particularly because it is unknown how robust the flood defenses are or how well the emergency response is organized in a real-life situation. There is also a scarcity of well-trained water managers, as well as a lack of knowledge transfer and societal awareness. The Hedwige polder in the Netherlands, along with an adjacent part of the Flemish Prosper polder, is the largest tidal area in Western Europe, covering 465 hectares. In function of the Sigma plan, the current Scheldedijk will be removed and replaced with a new levee further inland. The Hedwige Prosper polder is therefore being transformed into new tidal nature. Polder 2C's approach to the development of flood resistance measures is unique. The depoldering of the Hedwige Prosper polder has created a 6 square kilometer living lab environment where new and innovative techniques, processes, procedures and products can be tested for practical validation under very realistic conditions. Also implementing and successfully executing appropriate flood emergency procedures are critical to a successful flood emergency response. For this to succeed, you need the right kind of tools and expertise that is readily available. But how strong are our current flood defenses, and what is the impact of environmental elements such as the weather, the presence of vegetation, or man-made objects on our flood defenses? To answer these questions, numerous destructive field tests are carried out in the living lab. These dikes are currently getting extensively tested as climate change puts existing dikes to the test. Storm Corey recently demonstrated this where it reached the dike's outer toe. By doing this kind of test, it is thus possible to predict how such a dike will react after 100 years, say during a storm, and how much stretch remains in our system. Another essential purpose of the project is to gain a better understanding of how we structure our emergency response. Here we have a one-of-a-kind testing area to practice emergency management under controlled but realistic conditions. We intend to create tools and train levee guards in order to improve their current inspection methods. These are designed to test various levee restoration approaches under various conditions for major damages. Animal bows are particularly intriguing in this context, as no explicit consideration of animal-induced abnormalities is now made in dike design. This allows us to put several new recovery approaches to the test and evaluate if they are effective and have a place in the toolbox. The end of the project and the final breach of the Hedwige Prosper Boulder levee give a real-life event that we embed in a flood disaster scenario. This is a one-in-a-lifetime opportunity to investigate risk perception among citizens and decision makers. In this manner, we can prepare for large-scale crisis exercises across Europe and adapt our existing technologies accordingly. We also want to improve our knowledge infrastructure. Currently, expertise on flood defenses and emergency response is scattered and unformalized, and the lack of young, well-trained water managers is feared. The project's goal is to create a knowledge infrastructure via which existing and newly created knowledge will be made available, as well as to address the previously listed challenges in a toolbox. We also want to replicate our results internationally. The Hedwige Prosper Polder is an ideal place to conduct these kind of exercises and experiments, but we are, however, constrained by local levee geometries, vegetation coverage, and crisis management methods. We intend to connect our project outcomes to sites throughout the world by integrating them in international literature and state-of-the-art. A great variety of data is also being generated during the Polder 2C's activities. The Data Wizard is an online application tool that makes all the data available and simple to use for stakeholders such as researchers, levee managers, but also students for the future and time to come. Now several years in the project and the overflow and overtopping that's coming to an end, we can conclude that the existing dice can handle much more than what initially thought. But the good condition of our existing dice does of course depend on good management, regulated maintenance, and little irregularities in the design. Our dike inspection are also extremely efficient in Belgium and the Netherlands, but this needs to be further coordinated with foreign nations in order to build a unified inspection method and put it to the test in a variety of conditions on a regular basis. This toolbox will also provide these commonly agreed tools covering flood defenses and emergency response management, as well as specific topics like failure mechanisms, intervention approaches, and so on.
Hello everyone, I'm Astrid, a master student at CMET at the University of Ghent, and in the following 5 minutes I will tell you a bit more about my research topic, namely the role of the alkanivorax in the biodegradation of plastics in the marine environment. Plastics define the way we live today. We all use it and we all generate plastic waste. And this plastic waste can end up in the environment. In fact, up to 12 million tons of plastics are entering the oceans every year. And that's a problem, because the plastic degradation goes very slow, leading to accumulation. On the other side, also other contaminants enter the oceans, for example, crude oil. There actually exists a genus of bacteria called alkanivorax living in salty water and eating these alkanes. More specific, alkanivorax is degrading the oil when enough nutrients are available. Because plastics and crude oil have a similar structure, the connection between alkanivorax and plastics is easily made. And by performing 16S RNA gene sequencing of plastic degrading communities, this graph could be made. The x-axis represents the different sample names of the plastics enrichments and the y-axis the relative abundance. And indeed, we found alkanivorax in all tested plastic enrichments. But what is the alkanivorax doing with the plastic? What is its exact role? Is the alkanivorax degrading the plastic itself, or are other bacteria dominant during the plastic degradation and forming alkanes or other breakdown products? And is the alkanivorax using these products as a carbon source? First of all, we set up an isolation campaign for the different plastics enrichments, and after a couple of dilution steps we managed to isolate 2 species of alkanivorax, namely the TJ881 and the Burkomensis. We could isolate 2 species, but in reality there were also other species present in the plastic degrading community, as you can see on this graph, for instance the Alcanivorex hexenomutans. Alcanivorex are present on plastics, but are they active? This was tested by setting up an experiment in which a mix of cyclododecane and dodecane, nylon 12 or no carbon source were added to the Alcanivorex burkomensis isolates. And dodecane and cyclododecane can be seen as the structural building blocks of nylon 12, and that's why we made that combination. The incubation took 7 days in the dark. After 7 days, the concentration of cyclododecane and dodecane was measured with gas chromatography. The degradation products of nylon were analyzed with the TOC analyzer, and the flow cytometry was used to count cells. When we first look at the degradation of dodecane, we can conclude that it is significant in comparison with the control. The degradation of cyclododecane is not significant, but when we compare the 2 graphs, we can conclude that more dodecane is degraded. The alkane is more available as a carbon source due to its linear structure. When we compare the nylon degradation with the control, we can conclude there is no significant degradation. This can be explained by the fact that the nylon structure is too complex for Alcanivorex to break down in 7 days. This graph shows the cell concentration in function of the day for dodecane and cyclododecane, no carbon source, and the nylon. When comparing the growth on the dodecane and cyclododecane with the growth without carbon source, we can clearly see a higher growth when carbon source is present, and especially during the first days of incubation. This can prove that Alcanivorex really grow on the directly available structures. When comparing the growth on nylon with no carbon source, we almost see no difference. We might carefully conclude that Alcanivorex is not capable of degrading nylon in 7 days. So when Alcanivorex are not capable of degrading nylon alone, they maybe need other bacteria from the plastic degrading communities. So the following experiment will test the metabolic activity of the Alcanivorex in the community with plastics as a carbon source. And this can be done by using the cell sorter to sort the active fraction of bacteria from the non-active fraction, and to identify the different organisms, 16S rRNA gene sequencing will be used. I hope you all enjoyed my presentation, and I would thank my promoter, my tutors for the help, and if you have any questions, feel free to contact me. Bye!
Hello, I'm Steven, and I'm happy to present our research here to you about seasonal and spatial variability in determinants of phytoplankton biomass dynamics along a new offshore gradient. This study was a collaboration between Vliz, Kent University, and we were able to use the virtual environment of the Blue Cloud to do our modeling work. So the determinants of phytoplankton biomass are sea surface temperature, nutrients such as phosphorus, nitrogen, and silica. Further, we have solar irradiance and a top-down factor, which is zooplankton grazing on phytoplankton. Our study area is the Belgian part of the North Sea, which we divided into 3 regions, the near, mid, and offshore region. Further, we used data from LifeWatch and MeetMet Vlaamse, and this data was used to create daily input to drive our nutrient phytoplankton zooplankton model from 2014 until 2017. And the results can be seen here in this graph, where we have phytoplankton biomass by region. On the y-axis we have chlorophyll A, which is a proxy for phytoplankton biomass, and on the x-axis we have the time. We see that the highest biomass is observed in the nearshore region, even though the difference between the other regions may appear small. This is due to the fact that we are using a log scale, and the actual difference can be up to a factor of 10. Further, we observe that there is a two bloom pattern again in the Belgian part of the North Sea, with a high peak in spring, when spring bloom is happening, and a second smaller peak during the autumn. Continuing to the determinants and how they contribute to the changes in the biomass. So we focused on the relative contribution, which is the contribution of a factor divided by the total contribution of all the factors, and is expressed in percentage. And here on the y-axis we have the relative contributions on a scale from 0 to 1, so 0 to 100%, we can say that. And then on the x-axis we have the time. We have the 6 panels with the top row all nutrients, nitrogen, phosphorus and silica. The bottom row we have solar irradiance, temperature, zooplankton grazing. Here I want to highlight the temporal variability with the two main findings, that is solar irradiance and zooplankton grazing is actually an important contributor or an important determinant throughout the year, going from 38% up to 77%. Further, we found that there is a seasonal cycle going on. From autumn to spring, we have solar irradiance as an important determinant. As the day gets shorter, we only have like 8 hours of daylight, and that's important for the photosynthesis, so it starts limiting the phytoplankton. Then also during winter we have a decrease in temperature, also limiting the phytoplankton growth. So temperature plays a role in winter, but not during spring or summer. Then in spring we have the spring bloom, and that causes the nutrients to get depleted. First nutrient that gets depleted is phosphorus, followed by nitrogen and silica. There from summer onwards to autumn we have the zooplankton grazing that is the most important determinant, as the ratio between zooplankton and phytoplankton got shifted, and there's quite a lot of zooplankton compared to phytoplankton. Then we have the spatial variability, where we have clear near offshore gradient for the nutrients, where the nutrients are less limiting nearshore and most limiting offshore. We have riverine input from the Scheldt, and that gets depleted towards the offshore region, and so the nutrients become limited more in the offshore region. The opposite is true for solar irradiance, where we have that solar irradiance is most limiting in the nearshore region and less in the offshore region. That's because of the high turbidity in the nearshore region, blocking sunlight to penetrate the water. For temperature, we don't see a real difference, and zooplankton grazing is less limiting in the offshore region. So to conclude, we have simulated phytoplankton biomass dynamics, quantified the relative contribution of determinants, and found that there is a lot of spatio-temporal variability. And the outcome of this research could help to predict whether or not the ecosystem will change under future climate change scenarios and or economic blue activities. And with this, I would like to thank you for listening.
What is the impact of sand extraction on benthic ecosystem functioning? That is a question I'm trying to solve during my PhD, and today I will elaborate on some first findings. Sand extraction in Belgium started around 1976, and the demand for marine sands has been increasing ever since, especially for the beach depletions and the production of concrete for the construction sector. During such a sand extraction event, a large draghead up to 5 meters wide is swept over the seafloor and may remove sand up to 1 meter deep. This sand is then sieved on board, and only sand of a certain quality and grain size are kept, while the other particles are released back into the water. These two activities, on the one hand removing sediment and on the other hand releasing unwanted sediment back into the water, have an effect on the benthic biodiversity, the sediment characteristics, and also the seabed morphology. But what does this exactly mean for ecosystem functions and ultimately for ecosystem services and human well-being? Well, to unravel this, we compared the impact of 3 different sand extraction regimes on the carbon and nitrogen cycling within the sediments. To do this, we chose 3 extraction areas in the Belgian part of the North Sea. The first area is Noordhinder, which is characterized by a high extraction but periodic intensity, as these sands are mainly used for beach depletions, which happen in spring. Our second area, on the Tortonbank, is characterized by a continuous high extraction intensity. And finally, Ooststeek, which is characterized by a continuous but low extraction intensity. For these 3 areas, an impact and reference zone were chosen, and all zones were multibeamed to visualize the seabed morphology. After multibeaming, 3 to 4 boxcore samples were taken per zone. From these large boxcore samples, an incubation sample was taken and 5 smaller sediment cores. Once in the lab, these natural conditions were simulated within the sediment and within the incubation cores, and solute fluxes were also measured over time, such as the exchange of ammonium and also the oxygen consumption. From these smaller sediment cores, various sediment characteristics, such as the carbon and nitrogen content, were also determined, as also their vertical profiles. So over to the results. Here you can see multibeam data from one of our areas. It's from the Tortonbank. Here the tracks were most clear and on average up to 1 meter deep. Based on these maps and with the help of a high accuracy positioning system, we could also position our boxcore samples right within the tracks to really capture the direct impact of the sand extraction disturbance. Besides the multibeam data, we also measured the sediment permeability and also the sediment community oxygen consumption, which is a proxy for the mineralization of carbon within the sediments. As expected, the sediment community oxygen consumption showed a negative relationship with the permeability, as less permeable sediments will generally have a higher carbon stocks as they are better in trapping organic matter, while on the other hand, organic matter in the sandier sediment matrices will flow faster through. Even though a difference could be seen in permeability between the banks or the areas, all sediments remained permeable. Both for permeability and sediment community oxygen consumption, no real sand extraction impact was observed, only a difference between the banks. So to conclude, even though the impact on seabed morphology is very clear, no significant impact on sediment characteristics such as sediment permeability was observed. This limited effect on the sediment may explain that there is no impact on the first ecosystem function that was studied here, which is the mineralization of carbon, but more data on nutrient fluxes and element contents in the sediments are still coming in, which will allow us to draw more holistic conclusions. So I would say stay tuned for more. Before ending my talk, I would also like to thank Vliz and the crew of Simon Stephen for their help with the boxcore sampling and ship time, but also Niels for lending us their boxcore. Thank you.
The research vessel Simon Steven of Vliz is a multidisciplinary ship deployed for coastal and oceanographic research. And it's in operation since 2012, so that means it celebrates its 10th anniversary this year.
Providing services, both internally and externally, forms an integral part of the Vliz DNA. An important support service provided by Vliz is making research infrastructure available, including the research vessel Simon Stephen. The ship is owned and operated by DAB Fleet, while Vliz is responsible for the coordination of the oceanographic campaigns and research assignments. RV Simon Stevens serves academic coastal research in the southern bight of the North Sea and in the eastern part of the English Channel. It also serves as a training platform for students' marine sciences, as well as for maritime training courses, and as a testing platform for new technologies. The ship is equipped with both standard sampling equipment and sophisticated technologies, allowing scientists from various marine research disciplines in Flanders to study the atmosphere, the water column, and the seabed. To study the water column and its inhabitants, from microscopic planktonic organisms up to larger fish species, Vliz provides a whole suite of sampling nets and trawls. The lab facilities on board the ship enable scientists to process the catches and samples immediately. Vliz also uses the latest available sampling technologies. For instance, the Video Plankton Recorder, also known as the VPR, is an innovative underwater microscope that records images of plankton from a few nanometer up to a few centimeters in size. The VPR is an important tool within the European research infrastructure project LifeWatch. The Simon Stevens also acts as one of the seagoing carbon observation systems of ICOS, another European research infrastructure. To sample the surface and deeper layers of the seabed, Vliz makes different grabs and corers available. One such corer is the vibrocorer. This instrument allows researchers to collect and study 3-meter-long sediment cores from water depths up to 100 meters. Back on board, researchers can prepare the samples for storage in the core repository at the Marine Station Ostend or for further processing in the lab. The RV Simon Stevens is also equipped with a high-resolution multi-beam sonar. This device uses sound waves to visualize the soil topography and to detect objects in the water column. Researchers can, for instance, track wrecks of ships and airplanes on the North Sea bottom. Finally, the RV Simon Stevens can host and deploy the rigid inflatable boat ZCAT. The highly mobile ZCAT can be used for various measurements and samplings, and is the perfect carrier for a portable multi-beam sonar system. The ZCAT can be deployed from the shore as well for sampling in coastal waters, the Belgian ports, and the Schelde estuary.
Our next 30 minutes are devoted to a poster session and meet and greets with companies. Again, it's a digital poster session, so not the classic paper things we are all used to, but 1-minute videos. In a few moments, you will all leave this plenary session and you will be able to join different digital rooms. The posters that were selected for this Marine Science Day, they are divided in a few big topics, and each room covers one topic. There's also an expert there guiding you through the different posters, so you have poster topics, poster rooms that you can join, but also there are rooms for meeting and greeting companies, so you can make your decision there and explore half an hour. If 30 minutes, by the way, would be too short for you, no worries, because all posters, they stay available the whole day. You can also vote for the best poster until the end of the day. But instead of me explaining how everything works, please go out, explore as you are all used to do, and we will see each other back in 30 minutes.
ず君だよ。
Welcome back, everyone. I hope you enjoyed the breakout sessions and that you heard or learned something there that inspired you. If not, maybe one of the next 5 pre-doctoral researchers will inspire you, because we have again 5 pre-recorded talks now, where Annick Olivier, Dumas de Koning, Luna Geerts, Ilias Semori and Nille Meijers tell you more about their research. And remember, you can also vote for the best pre-doc presentation.
Hello everyone, from Nature Documentaries you probably all know the beautiful phenomenon whereby turbulence lights up the water with a bright blue color. Well, today I'm going to tell you a bit more about the organism that is responsible for this, and it's called Noctiluca scintillans, also better known as sea sparkle. But besides the nice light shows at night, Noctiluca can also be seen as red patches on the water column during bloom formation. Unfortunately, blooms of Noctiluca have been linked to increased fish and marine invertebrate mortality, affecting yields in fisheries and aquaculture due to its release of ammonia. Due to its adverse effects on the marine environment, we wanted to study how Noctiluca was doing here in the Belgian part of the North Sea. And therefore we studied its population dynamics, the drivers of its dynamics, and its impact on soft-bodied plankton species. For this study, we made use of 2 live watch datasets, one on zooplankton and one on various water quality parameters. Making use of zooscan analysis of WP2 net samples, the study was also the first one to present consistently counted Noctiluca cell numbers and cell length measurements. For the analysis, we eventually included 5 years of data on 9 onshore stations, which are represented here. And here we can see one of the first results. So there is a clear seasonal dynamic going on with high abundances of Noctiluca in May, June and July. We also see there is a really small population maintained in winter, and yeah, this overall trend is similar to what is observed in neighboring areas. But when we look at the dynamics of Noctiluca over the years, then we can see that there is an increasing trend going on with higher peak densities, and also that these peak densities occur more often. Also, the occurrence of Noctiluca itself in our samples nearly tripled in the 5-year period that we observed the species. So in 2014 it was present in 20% of our samples, while in 2018 this was already in 56%. So while our results suggest there is an increasing trend going on, we of course need more data to further evaluate and confirm this trend. And now that we know how the dynamics of Noctiluca are, we also wanted to know which environmental factors influence these dynamics. And for this we made use of a zero-inflated model, and it showed that temperature and phosphate were potential factors shaping the dynamics of Noctiluca. So previous studies in laboratories showed that the optimal growth rate of Noctiluca lied around 23 to 24 degrees. And yeah, this is in line with the summer temperatures that we observe here in the Belgian part of the North Sea. The zero-inflated model also showed that phosphate concentrations negatively correlated with Noctiluca densities, and we think that there might be an indirect link with food availability. So when the phosphate concentrations are low in spring, we think this is due to its consumption by phytoplankton species, which are commonly present, on which Noctiluca subsequently can predate and hence rise in abundance. And lastly, we also researched the relationship between Noctiluca and soft-bodied plankton species. We included Appendicularia, jellyfish, arrowworms and comb jellies in our analysis, but no negative impact could be observed. And the reason why we wanted to look at these specific relationships is because in neighboring areas, negative correlations and a really inverse relationship were observed. To conclude, we can say that there is a clear seasonality of Noctiluca in the Belgian part of the North Sea. It has high abundances in late spring and summer, and for you this means that this is a perfect time to go to our coasts and to search for the blue lights. And despite that Noctiluca is increasing and more frequently occurring in our data, no negative impact could be observed yet on the soft-bodied plankton community. We also want to say that anthropogenic pressures might play an important role in the occurrence and dynamics of Noctiluca, as temperature and phosphate were found to be the potential factors shaping the dynamics of Noctiluca. And then lastly, I want to give another small tip, and that is that you can follow Zeevonk Alert on Facebook or Instagram if you want to get notified the next time the blue light has been seen at our post. Thank you for listening, and my name was Anouk Olivier. I'm a PhD student and marine biologist at Vliz, and if you want to know more or have any other questions, you can always mail me at anouk.olivier@vliz.be. Thank you.
Hey everyone, and welcome to my presentation. My name is Dumas de Koning, and today I'll be presenting a method that we developed at ILVO to identify common sole in seafood products. And this method is faster, cheaper, and easier than ever to perform. As you all may know or may not know, seafood substitution, or the act of replacing an expensive species with a cheaper species, is rather commonplace. And usually this is done to make financial gains. So we speak of seafood fraud. I've listed a few major big studies and meta-analyses that showcase rather high rates of mislabeling or substitution in seafood on a global scale. And this has been going on for years with no clear sign of improvement. Unfortunately, next to economic consequences, this brings consequences for consumer health, ethical choices, and the environment. In Belgium, a study in Brussels took place a few years earlier that showed that out of all fish tested, 31% was mislabeled, with the top 3 most mislabeled species being bluefin tuna, Atlantic cod, and common sole. When we look at common sole specifically in this study, they noted that 11% was mislabeled. And then at ILVO, we later conducted another study in Flanders that had a larger sample size for common sole, which showed that 17% had been mislabeled. The problem is, once a fish is turned into a meal, it's no longer possible to identify the fish through traditional means. Luckily, analytical methods exist that allow us to identify a fish despite being processed. In Europe, isoelectric focusing and immunological methods are sometimes used, but most commonly we resort to DNA-based methods. DNA barcoding is the most commonly applied method because it's universal and it doesn't need to be adopted for each separate species. But it can only identify single species products and has rather lengthy and expensive protocols. So, how can we fix this? Well, we replace the commonly used lengthy lab-dependent commercial DNA extraction kits with a much faster but cruder DNA extraction by Tagliava and others, and combine it with loop-mediated isothermal amplification, or LAMP for short, since LAMP does not require a thermal cycler and is potentially faster than PCR. As such, we developed a LAMP assay that is specific for common sole. And then to visualize the LAMP reaction, phenol red can be added, which is a pH indicator that changes from pink to yellow upon amplification, which gives us a simple yes or no output. And to showcase just how easy it is and how we can apply this in the field, I'll be showing you a video of how we identify a common sole using kitchen equipment and basic lab equipment. Okay, let's see. I need to identify a common sole meal by tomorrow. That's easy. I'll just use the PCR. Oh no, looks like there's a power cut in the lab. Oh, I know. I'll just use a method that doesn't require the lab. So, to do this, we start by cutting a small piece of roughly 20 milligrams from the fish. We add the piece to a 100 microliter alkaline lysis buffer. And we crush the tissue using a micropestle. The lysis buffer is then heated to 85 degrees Celsius. And then we wait until the tissue is dissolved, which takes about 15 minutes. Then we add 300 microliters of tris hydrogen chloride to the lysis buffer and shake it briefly. We then take a tube with 39 microliters of water and a tube with 9 microliters of LAMP master mix, which is colored pink. We transfer 1 microliter from the lysate to the water to dilute the DNA. And then we add 1 microliter from this dilution to the LAMP master mix. We heat up the LAMP mix to 65 degrees Celsius for 40 minutes. And if the color changes to yellow, it means the food product contained common sol. In conclusion, we now have a fast, cheap, and easy to apply method that can identify common sol without requiring expensive lab equipment. Because there are differences in substitution rates between fish, it makes sense to actually gather a large amount of data for a single species. And that's where a method like the one presented here comes in clutch. It simplifies the entire process and makes it much more approachable. For example, the government could use this method to organize big audit campaigns for common sol. Additionally, the method can be adopted for other species by designing a LAMP assay for that species. Thank you for listening to my presentation. I hope you have a great rest of the day, and I bid you farewell.
Reducing our CO2 emissions alone will no longer be enough to limit global warming by 2 degrees Celsius. We also need to actively remove CO2 from the atmosphere. And in my research, I'm investigating whether this active CO2 removal is economically feasible. One way of drawing down CO2 is through the natural weathering of silicate rocks. However, this process is far too slow for us to rely on. Therefore, a new technique was developed, namely coastal enhanced silicate weathering, which is based on this natural weathering process, but essentially artificially accelerates these natural weathering speeds. A real-life implementation would look like this. First, we would mine the necessary rock needed, then grind it down to smaller grain sizes, then transport it by ship, and then spread it out into the coastal zone, after which the rock weathers and draws down CO2. And all these preparatory steps allow us to speed up this weathering process. For the mining of the rock, we would mine fast weathering rock, such as olivine. The grinding step allows us to expose more surface area to weathering. As for the transport and the spreading step, this allows us to spread olivine into optimal conditions, so that, taken together, we speed up the weathering process from a 1000-year timescale to a 10 to 100-year timescale. The economic feasibility, on the other hand, is investigated using a socioeconomic cost model. This is an equation that compares the application costs with the yields coming from coastal enhanced silicate weathering. The application cost is determined by the preparatory steps that I talked to you about earlier, and the yields is determined by the CO2 drawdown, but also by the profits coming from, or also by the CO2 credits. And the CO2 credits are essentially your price per ton of CO2. And the key takeaway from this equation is that coastal enhanced silicate weathering requires an initial application cost, but it earns us money back later. Looking at the per ton cost for 3 different grain sizes of olivine, we see that the grinding cost, shown in red, is the only variable cost, and that this overall cost, or the total cost, as a consequence, also becomes really large or exponentially large at small grain sizes. So looking at this graph, you might think, why would you ever use a small grain size like 10 micrometer if it's so expensive? Well, this is because the weathering rate is also highly dependent on this grain size. Using the same grain size fractions, we can showcase how fast they would dissolve. Here, I show you that the 10 micrometer grain dissolves within 100 years, whereas a 130 micrometer grain dissolves in 750 years. Using the same graph, we can also express this in the amount of CO2 being sequestered, and then we get this kind of graph. With on the y-axis CO2 sequestered per ton of olivine applied. Note that for all the 3 different grain size fractions, we would eventually sequester the same amount of CO2, but at vastly different timescales. Combining the costs now with the profits, we can calculate the profit curve for a 1 ton olivine application. And the one that I show here is for a 60 micrometer. Note that on the y-axis, I show you the cumulative earnings, and that this graph starts below 0, because we had an initial investment cost. Afterwards, this graph goes up, because CO2 is being sequestered, and also carbon credits are therefore being generated. Eventually, though, this graph flatlines, because all the olivine has been dissolved. So, What's a key takeaway point from this graph is that all the cumulative earnings are above 0 here, therefore we end up with a net profit. Comparing the 60 micrometer graph with the 130 micrometer graph, we find that the 130 micrometer grains are initially cheaper, however the total profits in the end are lower because they take so much longer to dissolve. Doing the same for the 10 micrometer graph, we see that we earn our money back faster. However, the initial or the much larger initial investment cost also makes it that total profits are much lower. So from this, we can derive that the 60 micrometer intermediary grain gives us the overall best profit. So in summary, coastal enhanced silica weathering seems cost-effective and promising from my analyses. However, before we can really apply it into the field, we require additional research still in terms of the ecosystem impact as well as societal acceptance of this technique. And with that, I would like to thank you all very much for your attention.
Hello, my name is Ilyas Samori, and I am going to present my work about the spatial-temporal dynamics in the gene expression of the copperpot Temora longicornis in response to environmental stressors. Copperpots are small crustaceans and are numerically the most prominent zooplankton group on Earth. They are critical to the function of ocean food webs because they are the major grazers in oceans and seas. Hence, they also play a significant role in the biological pump by removing fixated carbon dioxide in phytoplankton and taking it to the deeper water layers. Hence, copperpots help shaping the extent and pace of climate change. Yet on their turn, copperpots are affected by a complex combination of stressors, including those arising from climate change, eutrophication, and pollution. Therefore, monitoring programs have tried to quantify the link between environmental changes and the condition of marine ecosystems. In the Belgian part of the North Sea, dominant species found are the colonnade copperpots Temora longicornis, Acarcia clausi, Centroparis, and the harpactipede copperpot Euterpina acutifrons. However, comparing the densities of the colonnade species over time, we find a significantly lower density in the years 2015 till 2021 compared to the years 2009-2010. Densities of the harpactipede copperpot Euterpina are a notable exception. What could be the cause of these declines? Most zooplankton monitoring studies focus on biodiversity and biomass. However, such studies provide no information on changes in behavior, physiology, or changes at the molecular level. Since responses to environmental stress are initially genome-driven, we investigated the effects of changing environmental conditions and pollution on the gene expression of Temora longicornis. Zooplankton samples were collected with research vessel Simon Steven at 2 monitoring stations. More or less 40 zooplankton samples were collected at 11 different time points within the period 2018 and 2021. At each sampling station, conductivity, temperature, and depth profiles were collected next to other environmental parameters such as chlorophyll A concentrations. Additionally, we also determined PCB and pH concentrations, 2 classes of typically monitored persistent pollutants. From each sample, 2 times 25 adult copperpots were picked out on board and stored until they could be processed in the lab for RNA extraction. Sequencing was performed on a high-throughput Illumina NextSec 500 flow cell, generating 75 base pair single reads. Next, we tried to identify differentially expressed transcripts using the HR software. Next, we investigated several molecular functions and tried to correlate their expression patterns with the environmental parameters we measured. I will only present a few of them here due to time constraints. First, we looked at the expression patterns of differentially expressed phytologinin transcripts. Phytologinins are known to play a key role in reproduction, and several studies use them as a proxy for population density. In this plot, the Z-scores for all significantly expressed phytologinin transcripts of each sample are shown. A Z-score is a number of standard deviations by which the expression level is above or below the mean. Here we mainly see a significant upregulation in the February and June samples. We found a significant correlation between phytologinin expression and chlorophyll A concentrations, the measured summed pH concentrations, and the average biomass of the copepods, hinting these factors likely influence reproduction in Temora. Finally, we also looked into transcripts that have been identified to be effects in the crustacean stress response to heat stress and/or pH and PCB pollution. We identified 24 transcripts that were differentially expressed over time, mainly encoding for heat shock proteins. Many members of this group perform chaperone functions and are involved in keeping the cell stress under control. Significant higher expression of these transcripts was found in June samples. We found a small but significant correlation of these transcripts with temperature and the sum of pH concentrations. In conclusion, we investigated the transcriptome level profiles of the copepod Temora cornis collected in the BPNS over a 4-year-long sampling campaign. We identified significant expression levels for genes involved in metabolic processes and transcripts involved in the response to stressors. Hence, transcriptomics might be of aid to better understand the impact of the environmental on the physiology of zooplankton. Thank you for your attention.
Hi everyone, I'm Nana Meijers and I'm a PhD student working on microplastics at Vliz, Ilvo and Ugent. And today I will tell you about the detection and identification of microplastics in biota using NALRED and machine learning, validation of an innovative, cost-effective approach. So plastic pollution has become one of the most omnipresent threats the oceans are facing nowadays. Microplastics are of special concern as it has been shown that these are ingested by a wide range of marine species from different trophic levels. However, the environmental implications of microplastics ingestion remains poorly understood, which highlights the need for new, cost and time-effective methods. In this video, I will present to you Statio method, which we've developed recently and which allows for the semi-automated analysis of microplastics and mussels. This method was validated by spiking microplastics and mussels as part of the method qualification, and this validation will enable us to judge future measurement results. I will start by walking you through the sample processing step. So first, we spiked microplastics of known concentration, composition and size into mussel samples. Next, a two-step digestion was applied for each sample to remove as much organic material as possible. A 2-day alkaline and oxidative digestion was implemented with respectively potassium hydroxide and hydrogen peroxide. Samples were continuously heated and stirred on a magnetic stirring plate. In between both digestion steps, the sample was filtered over a stainless steel filter and sonicated to loosen all particles present. Next, the sample was filtered over a PTFE filter, stained with a fluorescent dye NALRED, and left to dry for 24 hours. Following this, each stained particle was photographed under a blue-green and UV filter using a fluorescent stereo microscope, which was then followed by image analysis. Here we can see each time the same spiked mussel sample photographed under the 3 filters, and both microplastics and some organic material is visible. As you can see, the particles present show a distinct fluorescence, and it is these differences in fluorescence coloration that allow us to distinguish plastics from non-plastics and to categorize according to plastic type. So to assess the quality of the method, we performed a validation. And to ensure good method qualification, we always worked with 2 people who each time calculate the validation parameters independently, and this based on their obtained recovery number for each sample. The mean of both will be shown here in the results section. A first parameter which we've considered for the validation of our method was recovery. For these experiments, it is expressed as the observed number of microplastics to the spiked number of microplastics. Next, we consider trueness. It is expressed as the degree of agreement between the obtained mean recovered number of microplastics for all samples and the reference number of spiked microplastics. It is influenced by the systematic error, which is also called bias, and here we calculated trueness and the bias B of the method based on 9 spiked mussel samples. We also considered the precision of the method during the validation, which is influenced by random error. And to do so, we first determined repeatability, which expresses the closeness of the recovery results obtained during the same run, but using different mussel subsamples. We also kept the measurement procedure constant and used the same equipment, the same analysis, and the same batch of reagents. Additionally, we looked at the within-lab reproducibility, which expresses the closeness of the recovery results, but this time obtained over 3 independent runs using different mussel subsamples and also using different reagent batches. Both parameters were calculated as the standard deviation S and the variation coefficient CV. Additionally, we also measured the robustness of the methods, which reflects its capability to remain unaffected by small but deliberate variations in method parameters, which in this case was sample type and mass. Here, we used 4 diluted and non-diluted muscle samples collected at the Belgian coast, and we also varied sample weight of 6 commercial muscle samples. Lastly, we looked at the selectivity of the methods, which is expressed as its ability to discriminate plastics from non-plastics, and also at specificity, which is expressed as its ability to discriminate different polymer types. To do this, we used our recently developed machine learning models and tested 20 particles. The plastic detection model can detect microplastics, while the polymer identification model can distinguish between different types of plastics. Both models were built using fluorescent color data from Nile red stained particles, and from these particles we extracted the RGB values and used those to train and validate our models. As input for the model, we used color data from our recovered plastics, and as output we got a predicted particle identity for each analyzed particle. Now onto the results section. So the obtained recovery for each sample was never below the set limit. The obtained bias B for calculation of the trueness was 17.31%, which, together with the obtained standard deviations and variation coefficients for the repeatability and the within-lab reproducibility, was within the set limit of 20%. For robustness, we obtained the mean recovery results, which also met the set criterion. Lastly, an accuracy of 100% and 90% was obtained for selectivity and specificity, respectively. So based on the obtained results of the method qualification, we can thus conclude that the newly developed method has proven suitable and reliable for the semi-automated, cost and time-efficient analysis of microplastics and metals. In the future, we plan to validate this method for microplastics analysis in fish stomachs as well, and we also aim to further automate the analysis part. Thank you very much for your attention.
And also thanks to these researchers for pre-recording their interesting talk for us. Right now it's time for a time-lapse video, and in this video you will see the construction of the brand new Innovation Campus. Every year, Vliz awards a number of prizes to deserving marine scientists. And one of those prizes is the North Sea Award to promote innovative research focused on the structure or the processes of the North Sea. And the 2021 award went to Emile de Borger, so congratulations for that, Emile. Besides, Vliz also awards two master thesis awards every year. Both fundamental and applied research in all branches of marine sciences is taken into consideration for that. And the two lucky winners for 2021 are Anton Bilsen and Merel van Gorp. Also, congratulations to both of you. Of course, these three people already knew that they were going to win because Emile, Anton and Merel, they recorded a talk about their research for us.
Hello all, I am Emile de Borger from the Royal Netherlands Institute of Sea Research, presenting my doctoral thesis titled Human and Natural Impacts on North Sea Sediment Biogeochemistry. But first of all, I'm very grateful to have received this award and this opportunity to present research that represents an effort of many people. So our research is focused on the seafloor, which consists of sediments, which are mud or sands, and is home to many different kinds of animals. And these animals feed on organic material such as algae, detritus, and dead organisms that drop down from the water column. And together with microbes in the sediment, this species community ensures that nutrients are recycled back to the water column. And so we get a lot out of the seafloor for free. So the biomass of animals on the seafloor sustains fish populations. So some species such as sole or plaice feed almost exclusively on organisms in and on the sediment. And as I said, nutrients are regenerated so that they are available to fuel the phytoplankton blooms at the base of the food chain. And the temporary storage of nutrients in the form of organic matter also prevents excess nutrient buildup in the water column. So it's a buffer to eutrophication. Recently, there's also been quite some interest specifically in how long carbon is stored on the seafloor, because if this is long enough, it could be counted as a carbon sink, so as a buffer for climate change. But as we've seen today already in previous talks, we humans do a lot on the North Sea which disturbs the seafloor. Dredging, the construction of offshore wind and other oil and gas platforms, and bottom trawling are all activities which have an effect on the seafloor and its inhabitants. So throughout my work, I first looked at how these nutrient recycling processes occur in the North Sea, with a focus on the Belgian part of the North Sea, which you see on the left. And so we did this because there is still quite some knowledge gaps about these seafloor processes, even in a well-studied system such as the North Sea. So any new insights are very useful for upscaling exercises and to increase our general understanding. Then we also need this info to align the current expansion of human activities in the marine realm, so the so-called blue economy, with sustainability goals and ensuring that we maintain a functioning, stable ecosystem. So for example, we also studied what the effects of bottom trawling and offshore wind farm construction are on the seafloor. I'll give a short example of offshore wind. So this is a map on the left of the Belgian part of the North Sea, where the current offshore wind farm developments are shown in purple. On the right is a graph of organic carbon recycling rates, these are on the y-axis, in relation to the coarseness of the sediment, which we measured in sediments in the Belgian part of the North Sea. So fine mud can hold a lot more organic material, because of its mineral structure, and therefore high recycling rates can be measured in these sediments. In coarse sands, on the other hand, a lot of the organic matter simply flows through, as water can pass through easily because of the coarse grains, so there is less organic matter to be recycled. But it's on sediments on this side of the graph where offshore wind farms are mostly constructed in our North Sea. So this is the case for the existing offshore wind farms, but also for the future planned expansions. Offshore wind farms are actually quite the hotspot of biodiversity, as the foundations of the underpart of a wind turbine are colonized by a whole lot of animals, and among which there is a high biomass of filter feeders such as blue mussels or anemones or small amphipods. And what these organisms do is they filter particles, so plankton and detritus from the water column, feed on it, and then deposit the remains as feces or dead biomass in the vicinity of the turbines, causing all kinds of changes. So we made a combination of models, a 3D hydrodynamic model and a biogeochemical model that includes this filtering effect by fauna on the turbines and looks at the effects of this on organic matter deposition, organic matter content in the sediment, and the processing of organic matter, which specific biogeochemical processes are used. And so what we saw as output was that after several years of operation, there could be quite the buildup of extra organic matter in the top layer of sediment. So local increases of up to 20% in contrast to the baseline simulation as a result of offshore wind farms. And so interestingly enough, we also saw a slight decrease of organic matter content a bit further away downstream from these wind farms. So this increase in organic carbon in the sediment transforms the sediments in offshore wind farms into much more active areas in terms of total organic matter cycling, with local rates increasing up to 60% in this case. So essentially transforming these areas into hotspots of organic matter cycling, where material appears to get concentrated by the activities of filter feeders at the cost of the downstream areas. Now, it's difficult to say how accurate these estimates are, since offshore wind farms are quite a recent phenomenon, so we're still following along what is actually happening nearby. And it's also not so easy to go there and collect a lot of sediment samples really close by. But with modeling tools such as these, at least we can identify knowledge gaps and anticipate what future changes may look like. So this is definitely true for offshore wind farms, but also for a lot of other blue growth activities, of which many are expanding so rapidly in recent years that our scientific knowledge is playing catch-up with reality. So, to remember from this talk is that marine sediments have a key role in global nutrient cycling and offer many, many more functions, especially in shallow coastal seas such as the North Sea. But these functions can all be altered by human activities. This has been so in the past, and it will be so in the future. And so for this reason, we need to keep studying this important but sometimes overlooked component of the marine ecosystem, especially in light of increasing use of marine resources. And so that was all that I could share in this time slot, but more can of course be found in the thesis and in associated articles. Thank you all for listening, and again a big thank you for everyone involved in this work and to Vliz for awarding this prize.
Welcome everyone, I am Merel van Gorp, and in this video you will learn more about my research about the hydrodynamic function of keels in bogfish. The body of bogfish is completely covered by a bony shell of fused hexagonal plates, the carapace, to which they owe their box-like appearance. As you can see, bogfish are excellent maneuverers. They can accurately control their body rotations, and they can easily maneuver themselves in and out narrow spaces during foraging. This is all quite remarkable, considering that because of their rigid carapace, bogfish can only rely on their 5 fins. With their unflexible body, they are similar to many rigid man-made aquatic vessels, where maneuvering, hovering, and precise landing at low speeds are strongly pursued. But even after decades of engineering research, they are easily outperformed by biological systems. Therefore, it's not unexpected that boxfish morphology has been the subject of several studies of bio-inspired design. The carapace is characterized by longitudinal ridges or keels. It was thought that these keels improve the swimming performance of boxfish in multiple ways. However, these hypotheses were never tested explicitly. In this study, we investigated which of the claims about the keels are true. 5 different boxfish species were studied, each of which had a different body shape. For each species, we compared the performance of a 3D model from a preserved specimen to a model where we digitally removed the keels. This was done by simulating the water flow around the 3D models, from which we then could calculate the performances. The first hypothesis was that the keels of boxfish would reduce the energy cost for forward swimming, as is the case in the leatherback sea turtle. This turned out not to be true for boxfish. The removal of keels had only a very small and inconsistent effect on the water resistance of the body. Furthermore, the keels were set to enable the boxfish to maintain straight swimming trajectories in turbulent water. This by passively correcting whenever the body is pushed slightly off course. This was claimed for up and down rotations and left-right rotations, similar to aerodynamic self-stabilization as seen in darts. However, recent studies found that this was definitely not the case. They measured that when thrown slightly out of balance, without active intervention of the fins, the body would keep drifting off. Considering that boxfish spend a lot of time maneuvering in their spatially complex habitat, a stable body wouldn't seem suited. Because permanent course stabilization would oppose every movement, this would make maneuvering energetically costly. From our results, we could confirm for all 5 species that the boxfish body was inherently unstable for nose up, nose down rotations and left-right rotations. No relationship could be discovered between the magnitude of the instability and the keels. These parameters were only very weakly affected by the presence of keels, and differences were inconsistent. The course of the water flow surrounding the body is very little affected by the removal of keels. The final hypothesis was that the keels of boxfish work similar to the keels in boats and ships, where they are used as a passive stability system to reduce the tendency to roll. This hypothesis, which was never tested before, was strongly supported by our results. All species showed a strong increase in roll resistance by the presence of keels. This demonstrates that the boxfish is less likely to be thrown off balance when it has keels. Now we know this, the next step is to find out how this improved roll resistance by the keels could contribute to swimming and maneuvering performance of the boxfish. This knowledge can in turn be used to enhance roll stability in engineered underwater vehicles without undermining maneuverability. Thank you for your attention.
Hello everybody. Today I would like to present you the work of my master thesis, which concerns the use of metabarcoding as a tool for intercepting invaders in the North Sea. Well, who are these invaders, you may ask? They are the non-indigenous species that are being introduced at ever-increasing rates into the world's seas and oceans. This is cause for concern because some of these non-native species begin to proliferate at the expense of native species and ecosystems, thereby becoming invasive alien species. And they are considered to be second only to habitat loss in threatening global biodiversity. They are especially problematic in the North Sea, which is considered one of the most heavily invaded ecoregions in the world. Examples of North Sea invasive alien species that you may know include the Japanese crab, the Japanese skeleton shrimp, the American slipper shell, and the Atlantic razor clam. In order to prevent even more non-indigenous and potentially invasive species from establishing viable populations in the North Sea, regular and efficient monitoring of the biota is required, especially in harbors, which are known to be introduction hotspots. There is a problem though. Most harbor biota survey protocols are based on morphological identification of the organisms. This approach is tedious, requires strong taxonomic expertise, and is prone to overlooking small or cryptic organisms. Therefore, more efficient species detection approaches are needed. To this end, 9 organizations, including the Flanders Marine Institute, have started the project GENES, or Genetic Tools for Ecosystem Health Assessment, in the North Sea region. This EU Interreg project, of which my thesis forms part of, aims to implement and standardize DNA-based species detection approaches. One such DNA-based approach is called metabarcoding. In this method, the total DNA is extracted from a sample and the particular gene or gene region, known as the barcode, is sequenced. Because the barcodes are in principle species-specific, they allow us to determine which organisms were present in the original sample. In my project, I collected 2 different types of samples. Using settlement plates, I collected sessile and epifaunal organisms, and using an Epstein net, I collected plankton. I then identified as many as possible organisms on the settlement plates using the morphological approach. I then proceeded to metabarcode the settlement plate samples using the 18S rRNA gene as a barcode. I did not attempt to morphologically identify the plankton, because this would have been extremely difficult and given few meaningful results on account of the small size of these organisms. Instead, I proceeded to metabarcode them directly using 2 different barcodes. The sequencing of the DNA was performed using the state-of-the-art nanopore method. In this method, a single strand of DNA, driven by an electrical current and aided by an enzyme, passes through a nanopore on a membrane. As it does so, each of the 4 different nucleotides within the strand reduces the current across the pore to a different extent, and these nucleotide-specific current drops are measured by an electrode, thereby allowing the DNA sequence to be read out. Because nanopore sequencers do not rely on imaging equipment, they can be very compact and portable, as demonstrated in this picture. In addition, they are capable of sequencing quite long strands of DNA, which increases species resolution, and they also exhibit a decreased sample preparation time relative to traditional sequencers. So now I will go over the results of my study. From the settlement plates, I was able to identify 14 taxa to species level using morphological examination, and over twice that amount of taxa using the metabarcoding approach. However, I was able to find only 2 species using both methods. From the plankton samples, I was able to assign 21 and 27 taxa to species level using the CO1 and 18S barcodes, respectively. Again, only 2 species were found by both methods. Metabarcoding enabled me to detect the presence of several species that were not previously recorded from the Belgian part of the North Sea, but which are known to be troublesome invaders in other temperate marine areas. These include the oyster parasite Polydora haswelli and the invasive copepod Oitona davisi. So, what can we learn from the results? Well, I demonstrated that metabarcoding has several advantages over the morphological approach. Metabarcoding enabled me to discriminate between morphologically very similar species and it also identified a whole array of small and cryptic life forms, such as plankton and larvae. It is often these tiny life forms that are most apt at spreading and invading new areas. In addition, I demonstrated that nanopore sequencing is very time efficient. However, I also noted that metabarcoding has some challenges associated with it. As I mentioned, only a few species were detected by more than one method, and this low agreement may be caused by incomplete reference databases, false negatives due to technical biases, and differences among the barcodes. However, these challenges were often related to the recent implementation of metabarcoding as a method, and they could be ameliorated to some extent by combining multiple species detection approaches. So, what's the overall conclusion? Metabarcoding is certainly a promising tool for detecting species, both native and non-native. Thank you very much for listening to my presentation, and I hope you enjoyed.
In 2021, Vliz also honored 4 brilliant marine research ideas with a prize. And 3 of the winners are here live with me today, and we will have a chat about life and work. And yeah, my first guest is Soria Delva. Soria, welcome.
Hi.
How are you?
I'm really well, thank you.
Looking forward to this talk?
Yes,
okay, cool. Soria, to people who are not familiar at all with what you do, how would you explain your research in a few simple sentences?
Yes, so I'm investigating DNA methylation in a brown seaweed. Okay. Usually when we think about DNA, we think about a sequence of 4 bases, A, C, T, and G. But there's also different molecules that can bind to these bases. And one of them is a methyl group that is often added to the C, and this is what we call a DNA methylation. And it's gathered a lot of attention lately because it's involved in the regulation of gene expression. Usually it's been thought of as this big silencing mechanism, so it switches off genes. In truth, it's a bit more complex than that, depending on where it occurs and so on. But it's true that it's a very important mechanism for a lot of cellular processes.
And why is it important in brown seaweed?
In this case, we're really interested in it because not a lot is known about it. So when it comes to the seaweed world, we're really lagging behind. To date, there's only 1 methylome available for a seaweed species. And other than that, we don't really know much about it.
What's the reason for that? That they were not interested in seaweed for decades?
No, actually it has to do with a study quite some time ago in Ectocarpus, where they thought there was no DNA methylation in that seaweed. Oh. And Ectocarpus is kind of a model species in the seaweed field. And because with the techniques they used then, they couldn't find any indication of methylation, they decided.
And recently they found there is DNA methylation, and
exactly.
You studied why exactly?
In my research, we're mainly interested in the inheritance of methylation patterns from parents to offspring. So we want to know if parents can somehow transfer some information to their offspring via DNA methylation.
Information also about the environment they live in?
Yes,
and
exactly.
That's important in these times of climate change, I can imagine?
Yes, exactly. So for us, it would mean a very huge deal, especially with all the changes that are going on in the ocean. An organism basically has 3 ways to respond to that. Either they can move to more favorable conditions, or they can adapt to the changing conditions, or they die. And especially the adaptation part, there it's very interesting to know if DNA methylation could play a role in adaptation on the short term, because it would allow for parents to transfer some information about their environment to their offspring. So let's think about it in this way. If parents would be encountering a certain stress, and they would be able to transfer that information to the offspring, which could make the offspring more resilient, would be very important in this kind of...
And I think you brought parents and offspring, right?
Yes, exactly. So this is a female seaweed.
Female?
Yeah. Yes.
Can I see?
So, yes.
Is it a female or a male?
Yeah, it's super small, but you can see the eggs on the bottom.
You mean this side?
Yes, yes. So there you see the dark patches, they're eggs. And also on the seaweed itself, you can see at some places some black patches, and these are eggs that are
and the males
produced.
don't have eggs?
No, the
Okay.
males produce sperm cells.
Okay, and these are the offspring.
And these are the offspring, yes. So
Really
these are
small, right?
baby algae. Yeah, they're only 2 to 3 weeks old, so they're still very young.
And how long does it take for them to be a big seaweed?
A couple of weeks, definitely. Yeah, even after this, still a couple of weeks before they will attain quite a big size.
And these are the things you are working on
Yes.
every day.
Yes.
Sonia, climate change, you mentioned it, has a big influence on the work you do. Are you optimistic or pessimistic about our future?
Yeah, it's a very
You can
difficult
be honest.
question. Yeah, I think I'm a bit afraid of what's about to come, because we can already see quite some changes in the sea.
In the communities of seaweed?
Yes, yes.
Really?
So yeah, there's already, in some places, there's already a whole shift in the algal communities. There's species that are disappearing and that are moving more northward. So I'm a bit afraid of what is still to come, but I'm also hopeful that we can still do something about it, and hopefully we find a way to fix all of this. But yeah, I would say maybe a bit pessimistic, but with hope.
With hope, and
With hope.
hope is a good thing. I wish you
Yes.
all the best of luck with your research, and thank you very much for being here.
Thank you very much for the opportunity.
It's a pleasure. The second winner of a Brilliant Marine Research Ideas Award, that's a difficult word, in 2021 was Alexander Hooijberg. Alexander, welcome.
Hello.
Hi. You brought some kind of medical device, or
Yes,
not?
that's true.
What did
well,
you bring?
This is actually what we bought with the BMRI grant.
Oh.
And it's the Nexus 10 Mark II of Mind Media. A difficult question.
Mm-hmm.
And what it does, it measures the stress response of a human being. If you are stressed, there are a lot of changes happening within the body.
Yes.
And it results in a preparement of the body to prepare to fight or flight,
Okay.
or to rest and digest. And by measuring these symptoms, we can actually get a view on the internal processes of a human being. So it's a lot used in medical research, of course.
And what does it have to do with the sea?
Well, we want to investigate whether the sea has an impact on our stress, or the reverse, or relaxation.
Alright. And did you already find something?
Yes, we found that it's still mainly a perceptional thing, so we also measured subjective variables.
Yes.
But with this device, we have one of the first evidence that there is also changes in the respiration rate, for example, so that people, when they are exposed to the coast versus an urban environment, that at the coast they breathe slower compared to at an urban environment.
But first signals, not enough already to make it hard, or?
Yes, well, there is a lot of data. You see, there are a lot of sensors here, and they each have to be artifact corrected, because people are moving, the measurements are not very pure, so we have to do a lot of pre-processing, and that's still a bit ongoing. But we also calculated the direct measures that came out of the device, and from there we see this increased respiration rates, but also increased brain state or brain activity, for example, of the frontal lobes.
Okay.
And that is indicative for that the environment needs to have, or needs to be processed more by the brain, and more specifically urban environments are more
because of more things going on.
More distracting, more difficult to process.
And why is it so important to do this research? What do you want to prove with it, or change with it in the world?
Indeed. Well, a lot of people say to me, yeah, we already know that the coast is healthy, why should we do more research about it? But we also know that a lot of people experience stress and other mental health disorders such as depression and anxiety, especially now during COVID times. And increasing knowledge about the internal processes of the body, how it responds to the coast, can actually give us the necessary knowledge to use the coast as a therapy.
Wow.
If you can, for example, there are a lot of general practitioners which receive a lot of mentally ill people. If they can prescribe the coast to those people, it would provide a very cost-effective solution, practically free, and it would also benefit the blue tourism sector, of course.
Sure. Sounds like science fiction, but it's really science.
Indeed, yeah.
Alexander, imagine you would have an unlimited amount of research money. What would you like to do with it?
Difficult question. Very difficult. I would, enlarge my team. I think that's the main issue, because now we have so much data, for example, from this device, so much ideas about future studies that are very relevant in today's context.
Do you have an example?
For example, a real-life experiment is also on the planning that we are doing. And we can always use a lot of help, and help costs money, of course. So that would be very fun to do, to really make this field of research a bit more, to make us larger and make sure that we really can provide the science that we need for the ocean interactions that we want.
Well, I wish you all the best of luck with that. Thank you very much for being here.
Thank you as well.
And the third winner of our brilliant marine research ideas is Valérie Matelin. Unfortunately, Valérie could not join us here live today, but if we learned one thing during the pandemic, it is how to set up Zoom calls. So Valérie joins us by Zoom. Hi, Valérie, can you hear me?
Yes, I can hear you.
How are you today?
I'm fine, thank you.
Well, you won an BMRI award. Your research is about microbial communities and plastics. How would you explain what you do in a very few simple sentences?
So I'm actually looking at novel plastics, and we're looking at how these plastics could be degraded by microbial communities that are already present in the seawater.
What are novel plastics?
Novel plastics, it's plastic that are still also being designed. Let's say I work together also with a company that is a polymer architecture company, and they design novel plastics, so new plastics that are not yet on the market, but we are investigating their biodegradability in the marine environment.
And will we use these plastics as consumer things, or is it more industry-wise?
Well, the company has several projects running. I'm not very sure what the outcomes of those will be. I'm really focused on making the plastics biodegradable, but I have no idea of their customers.
And what are your main results until now from the research you do?
So we see that when we bring the plastics in the microbial community of seawater, that this community develops to a new community, or a community with a different microbial composition, which can indicate that several groups of microorganisms have the ability to degrade those plastics.
So they are still somewhere in the ocean, but you see them grouping around the new plastic. Is that right?
It's lab scale experiments. We're not throwing plastics in the ocean, but on lab scales you can see that really some microorganisms are interacting, let's say, with polymers.
Mm-hmm. Let's say 20, 30 years from now, what do you hope that the research will have achieved?
So I really hope that we will be able to find a good way to, or a good protocol, let's say, to design polymers that can be biodegradable once they end up in the environment, so we can prevent the massive accumulation of plastic waste.
All right, I wish you all the best of luck with that, and thank you for being here. Bye-bye.
Bye, thank you.
last winner of a BMRI award was sponsored by Deme and Antea Group, by the way, is Glenn Stripsteen. Welcome, Glenn.
Thank you.
How are you?
I'm really good.
You're
I'm
really good.
Happy to meet you in person.
Oh wow! Okay, Glenn, I'm really intrigued by your research because you build with nature.
Exactly.
What is building with nature, and why do you do it?
So my research focuses on a really unique pilot site. So we basically planted marram grass, what you see here, on the upper beach in front of the traditional dike, and we used that ingredient to develop a dune. So we have like 3 ingredients that we need to develop a dune. We need wind, we need sufficient amount of sand, and we need marram grass.
Wow, you're good
and
here.
the combination, we are really good here, yeah, exactly. So we use this kind of solution to develop a dune, and the first place why we installed this dune is because we have a lot of problems on the dike. During high wind events, there's a lot of sand transport on the beach, and a lot of that sand ends up on the dike and on the promenade.
Oh, yes.
So it costs a lot of money for the government to clean that up. And basically, when we have this dune in front of the dike, it captures all the sediment coming from the beach. And the positive side is that we develop a dune, so it's really positive for coastal protection.
It sounds really like something, yeah, from the older times. I mean, building with sand and wind.
Yeah.
Why did we not think about this much earlier?
It's a really good question. Like in the beginning, the Belgian coast was consisting a lot of natural dunes, and then in the storm of the 50s, there was a really large storm with a lot of casualties. And then we started to develop, like, we worked a lot with concrete, so we built a lot of dikes and walls to protect us, but that's not good enough for on the long term. And now the Belgian government is changing to a more natural solution, using marram grass to develop dunes, and that's really good for coastal protection.
Do storms like the ones we had a few weeks ago, Eunice, Franklin, do they affect what you do, the dunes?
Yeah, so we have basically 3 pilots now, pilot sites now, and the one in Oosterhoeve really close by here. We had a little bit of dune erosion in front of the dune foot, but not a lot. But we saw in the 2 weeks after that storm, the dune was already recovering.
Wow.
So new sand from the beach was ending up in the dune again. So it's really resilient. The one in Raverstede did not have any dune erosion, so during these storm events, we did not see any erosion, so it's really resilient.
Oh, cool. I was also wondering, to implement these kind of things, I mean, research has to be communicated to people making decisions about, okay, not building any apartments anymore, investing in dunes. Is this, in our country, something that goes well, that information from research to politicians, basically?
It was not, but now we see a change. So that information, since we do a lot of science now on these kind of solutions, this is communicated well now towards the government and other instances, and we see that information is receiving quite well. So we hope in the future that we can build more with these type of solutions to protect us.
Cool. Yeah. What is the yellow thing you brought?
The yellow thing is a controller from a GPS. So we did a lot of measurements. Basically, we go to the beach every week with a GPS, and we measure the topography within the dune to see how the dune is developing.
Like length, height? No.
Yeah, X, Y, Z coordinates,
okay.
And based on those that information, we can calculate volume changes within the dune, and we see within 1 year that the dune grew with 14 cubes per meter per year. So that's about 1 meter in elevation.
Wow.
Yeah. So that's quite good. So in the last year we saw a really big development of the dune.
I always thought dunes was something of 20 or 30 years, but even in 1 year you can achieve something that has a scientific
exactly.
impact.
As long as you have the ingredients, the 3 ingredients, marram grass, wind and sand, you can develop a really nice dune.
Well, thank you, Glenn, for being here, and good luck with your research in the future. Yes, Valerie, Alexander, Soria and Glenn, they were the brilliant marine research IDs, or in short BMRI winners of 2021. But yeah, a new year has come, we're already 2022. So it's my pleasure to announce 6, not 4, but 6 new BMRI award winners today. The first one is Wouter de Leersnijder from Ghent University and KU Leuven. His award is co-sponsored by the Antea Group, by the way. The second prize goes to Injas Pelkmans from University of Antwerp, co-sponsored by DEME. The third award winner is Marie Sioen from Ghent University and University of Antwerp. Her prize is co-sponsored by Othari. The fourth prize goes to Hisham Shaikh from Vliz and Ghent University. And then we have 2 more prizes for people of the University of Antwerp. First of all, Helene Kersenbelk and also Jesper van Dijk. So I would say congratulations to all of you. Spend the money well in the coming year. And as you know, yeah, you have to come and talk about your research next year here with me. So good luck and see you next year. And finally, we also have the winner of the Delcroix Award with us, Marine Séverin. Welcome. By the way, Delcroix Award, that's a prize awarded by Vliz and Hydro to stimulate research around oceans and human health. Marine, hi. I talked to Alexander a few moments ago. You also research oceans and human health, but what do you do specifically?
So I look at specifically the psychological mechanisms that explain why the coast has a positive effect on our mental well-being.
Okay.
And I specifically look into the emotions that we feel at the coast and how these emotions improve our well-being.
And how do you do that? Because Alexander measures heart rates and respiration. How do you measure emotions?
Well, so there are different ways, but one way is that I did a qualitative study, and that basically means that I did interviews with a group of coastal residents, and in those interviews we asked them what they feel when they're at the coast. So that's one way to look at emotions, and another way is to use questionnaires that allow us to measure their emotions.
But that can also be rather subjective, I guess. How do you filter out for these?
Well, I mean, emotions are always subjective, but we always answer it on a scale. So from 1 to 7, how much do you feel that emotion? So some people might feel it more than other people, for example.
And what are your main results so far?
So far, we see that when people are at the coast, they feel this emotion that we call awe.
Oh.
Yes.
Okay.
So it's spelled AWE, and it's kind of like a wow effect. So when you're at the coast and you look at the horizon, and usually you feel that you are in the presence of something bigger than yourself. So this makes you feel more small and maybe a bit more humble in life. So that's one emotion. And another emotion that we look at is that people feel nostalgic when they're at the coast. So they sometimes think back to their childhood or the different memories that they had when they were there. And finally, we also find that people feel more connected to nature when they're at the coast.
Of course. Why did you decide to do this research? Did you grow up by the sea?
No, I didn't, but I always enjoyed going to the coast, and I've always noticed that when I went there, it made me feel better and happier and more peaceful. But yeah, there must be a reason for this, and emotions are very important for our well-being, and understanding them helps us understand better what makes us feel better. So I wanted to look more into that.
Wow. Yeah. If we think about emotions and nature, I think the pandemic, the COVID-19 pandemic comes to mind. Did you study something concerning the virus or not?
Yeah, so the COVID-19 pandemic we kind of took it as an opportunity because it meant that the majority of the Belgian population didn't have access to the coast anymore. And that only people who lived near enough had access to it. And so we wanted to see whether there was any difference in well-being and also in the emotions that we feel when we would go outside. And we found that people who did have access to the coast, they had a higher well-being and they felt less bored and less worried, and they were happier than people who didn't have access to the coast.
Wow.
And we always controlled for typical factors such as socioeconomic status and mental illness, and even after controlling for that, we found a significant difference.
Wow, that's really, I didn't know. Even I grew up by the coast, but I didn't know. Imagine you could, go 20, 30 years further in time. What do you dream of? What are your main projects for the future?
Personal projects or projects regarding the research?
Both.
So I think, I'm a clinical psychologist, so I would like to go deeper into therapy and also, just improve my maybe clinical psychologist skills, but also continue the research, of course, and maybe look into how these emotions might differ according to different population groups, so different ages or different socioeconomic status. I hope to continue this research.
Cool. I think the combination of both is really cool. Well, good luck with all the things you want to do in the future, and thank you for being here. I have your official prize here. It is the Delcroix Incentive Award, so that's yours. Well deserved. Congratulations.
Thank you very much.
Thank you. Yes, we all know that scientific research is impossible without the help and support of our government. So that's why Hilde Krevits, she's Vice Minister President of the Flemish government, but also Flemish Minister of Economy, Work, Innovation, Social Economy and Agriculture. She has a message she wants to share with us.
Dames en heren, beste onderzoekers van de zee. Ik ben bijzonder verheugd om jullie te mogen toespreken op de jaarlijkse hoogmis van de marine wetenschappen in Vlaanderen. Liever had ik jullie recht in de ogen gekeken en de hand geschud, want niets is leerrijker dan een face-to-face ervaring, omdat je dan nieuwe ideeën kunt ontdekken, nieuwe inzichten kunt krijgen of kennis kunt delen met andere mensen. En laat nu net onze zee met haar boeiend ecosysteem en de ruige krachten een onderwerp zijn waar veel over te vertellen valt. Nieuwe cijfers tonen aan dat onze Noordzee elke plek voor gemiddeld 4 gebruiksfuncties laat aanwenden. We hebben allemaal een band met de zee. Zij het als kusttoerist in het mooie West-Vlaanderen voor het vinden van rust of ontspanning, of aan de keukentafel wanneer de pot een tong platijs of misschien wel een tomaat crevet schaft. De zee is een onuitputtelijke bron voor voedselproductie. En als we er zorgzaam mee omgaan, dan kan dat nog lang zo blijven. Daarnaast hebben ook veel mensen hun job te danken aan de zee. Denk maar aan onze havenarbeiders en de bemanning van vrachtschepen. Of jullie, de Vlaamse Marine Onderzoeksgemeenschap. Maar het gaat eigenlijk nog veel breder dan dat. De zee biedt ook ruimte voor de opwekking van groene windenergie. En dat alles maakt onze zee meer dan ooit maatschappelijk belangrijk. Elk van ons heeft er dan ook alle belang bij om verantwoord en duurzaam met onze zee om te springen. De bron van die duurzaamheid is kennis. Beste onderzoekers, Vlaanderen en ver daarbuiten heeft jullie excellente marine kennis nodig. Kennis is de basis om de grote uitdagingen die voor ons liggen aan te pakken. Het doet mij dan ook een groot plezier dat ik vandaag de nieuwe kennisgids van het Compendium voor Kust en Zee mag voorstellen. In deze gids heeft het Vliz samen met meer dan 100 experten een pak informatie en cijfers gebundeld over het gebruik van onze kust en onze zee. Het verschaft een uniek zicht op onze marinekennis. Deze 4e editie maakt meteen duidelijk dat we al ongelooflijk veel weten. De marinewetenschap is er de laatste jaren met rassenschreden op vooruit gegaan. Het stemt me gelukkig dat projecten met een actieve betrokkenheid van ondernemers, het beleid en maatschappelijke groepen nu stilaan de norm zijn. Dat ook burgers de laatste jaren actief bijdragen, is de kers op de taart. Ik wil daarvoor uitdrukkelijk het Vliz bedanken. Burgerwetenschapsprojecten zoals de grote Schelpentaaldag en Sea-Watch B zijn een groot succes. Jullie laten op die manier de brede bevolking op een toegankelijke wijze kennis maken met wetenschap en laat net dat nu een van de streefdoelen zijn van mijn beleid. Beste mensen, met Vlaanderen grijpen we dit decennium van oceaanwetenschappen aan als het momentum om belangrijke stappen te zetten in maatschappelijk relevant marien onderzoek en om onze groeiende kennis nog meer om te zetten in duurzame oplossingen in de praktijk. Een versterkte samenwerking tussen overheden, burgers, wetenschappers en ondernemers is cruciaal. Alleen samen kunnen we uitdagingen op het vlak van klimaat en biodiversiteit het hoofd bieden. Het Vlaamse Instituut voor de Zee doet haar naam hierbij alle eer aan, door een centrale rol op te nemen in het bevorderen van die samenwerking. En dat is goed. De nieuwe, prachtige InnovOcean campus, waar ook het ILVO zal huisvesten, wordt daarvan de fysieke bekrachtiging. Samen is de enige weg vooruit. Dank jullie wel en ik wens jullie ook alle succes toe.
Did you know that, on average, every point in the Belgian part of the North Sea is claimed by 4 different user functions? Belgium disposes one of the most intensively used strips of coast and sea in the world. Luckily, we are able to match that with a significant volume of scientific knowledge and expertise. Yet, with all these different users, keeping an overview of the relevant scientific publications, reports, policy documents, guidelines and legislations and so on might give you a little headache. Maybe you are struggling to find a competent authority for dredging activities in the North Sea, or you desperately need the most relevant scientific publications on the impact of underwater noise on the marine environment. Well, the knowledge guide of the Compendium for Coast and Sea might be just the right remedy to your issues.
The what?
The Compendium for Coast and Sea is a one-stop shop for all information on the coast and sea in Flanders and Belgium. The knowledge guide showcases the most relevant scientific publications, government reports, socio-economic figures, current legislation and so on. We do have to warn you, however, the compendium holds a lot of information. So don't just start reading yet. We will show you how to use the knowledge guide. Every thematic chapter has a comparable structure. We start off with a short introduction of the user. This is followed by the policy context, spatial use, societal interest, impact and sustainable use. Also, every chapter ends with the legislation reference list. So let's put it to the test. Suppose you need figures on the employment in the Flemish ports. Well, you take the chapter on maritime transport, shipping and ports, and you go to the section on societal interest, and there you have it. Oh, and did we already mention that all hyperlinks in the knowledge guide lead to the source material and more in-depth publications, which by the way are almost all openly available in our online library? Or perhaps you want to know which zones in the Belgian part of the North Sea are reserved for military purposes? Let's scroll to the chapter on military use and go immediately to the spatial use section. Here you find all you need. Also, you can immediately link to our coastal portal. The coastal portal provides users access to spatial information and data related to the functions and activities that take place in the Belgian part of the North Sea and the Belgian coastal zone. It's pretty simple and convenient, isn't it? But of course, you don't have to take our word for it. Do try it yourself. The Flanders Marine Institute is coordinating the Compendium for Coast and Sea, but we could not have done this without our network of over 100 experts from science, policy, industry and societal organizations. Therefore, the Compendium for Coast and Sea can be regarded as a co-creation effort of Belgium's entire marine landscape. It is clear that the Compendium for Coast and Sea puts a wealth of marine information at the tip of your fingers. We do believe that this brings us one step closer to achieving the main objective of the UN Decade of Ocean Science for Sustainable Development. You want to know more? Contact us at compendium@vliz.be or feel free to visit our website.
And with this nice visual overview, we're nearing the end of the Vliz Marine Science Day 2022. He opened the conference or the research day, and he will also officially close it. So please welcome back Jan Mees. Jan, how did you experience this second digital Marine Science Day?
Although it's digital, it still gives me a lot of energy.
Okay.
I'm very much looking forward to the physical event again, hopefully next year.
Yes.
But this being said, I'm impressed, like always, by the diversity of what we have seen, the quality of what we have seen. A small region, but it has a lot to offer with regard to marine sciences, and that's something that always makes it worthwhile for the whole Vliz community, I think, to appreciate what we are doing and why we are doing it.
Cool. If you would have one take-home message for all the people behind their screens, what would it be?
Stay involved, stay connected through Vliz to the global ocean research priorities. We can channel this for you. We will have a lot of exciting activities in 2022, opening a new building, opening a new ocean innovation space, celebrating 10 years of our research vessel, the Simon Steven, and a lot of other activities. Come to our events is something I want to say, because we want to channel all this excellence towards international networks, basically.
So stay involved, because we need you.
That's it.
That's it.
Yes.
Okay. Thank you, Jan, for being here today, but also for being one of the driving forces of marine research in Flanders, but also the rest of the world. Thank you very much for closing the Marine Science Day. Yeah, well, then I think it's officially time to wrap it up. It was an honor to be your host today. Yeah, please remind that all posters, they stay available until the end of the day, so you can vote and for the best poster and for the best pre-doc presentation. I talked about a cool gift, so this very nice Vliz t-shirt, that's what you can win, some of you, if you vote, so please do that. I would like to end by thanking the organizing committee, thanking the video and the sound crew also, and all people involved in making this day happen. And of course, I would like to thank you for being here with us today. Please keep up the good work, stay safe, stay healthy, even in these difficult times. Thank you very much, and have a very nice Wednesday. Bye-bye.